Okay, this is a brief explanation of the process of replication for honors biology. So for starters, replication is the process of copying the DNA, and this happens before the cell is going to divide, because every time there's a new cell, it needs to have a copy of the DNA that's there. So at first there was a lot of debate about how the DNA actually copied itself, and there were three possible types of ideas for how this could happen. There is what we call conservative replication, which was basically where the old strand would stay completely together, exactly the same, and there'd be a brand new strand made. Another possible idea was where the DNA would come apart and the two new strands would be made of different parts, some of them old, some of them new, and it'd kind of be a mix and match. And then the last possible idea was what they call semi-conservative replication, which basically means that the DNA would divide in half, and half of each old strand would be part of the new strand, and then there would be one new strand of each of the new. So each was half and half, but not so mixed up. And what it turned out to be was that replication was semi-conservative, where basically our DNA is getting split in half, and there are two new strands being formed as this is happening. Now this is a really simplified diagram of what's going on, but we're going to use it to kind of explain the basic steps here. And there are a lot of enzymes and a lot of proteins that are involved. So we're going to kind of take it step by step. Now before we can really get into this, it helps to remember what the structure of DNA is like and what exactly is going on with the sugars and the phosphates down the side. As we talk about this, I'm going to be using the numbers 5' and 3'. And basically what that's referring to is the carbons that are in each of those deoxyribose sugars. They were each a 5-carbon sugar. Now if we were to start numbering these, starting at the carbon that connects to the base, that would be 1, 2, 3, 4, and here's 5 connected to my phosphate. Or this one, if I start counting clockwise, we'd have 1, 2, 3, 4, and here's the 5 connecting to the phosphate. So you can see that depending on which phosphate we're looking at, it's either connecting to the 3 or it's connecting to the number 5. And these run opposite. So down here, the bottom carbon on the sugar is the number 5, our 5' sugar. Whereas here, it's my number 3 carbon. So this is my 5' end, this is my 3' end. Now if I were to go and compare that up at the top, you can see it's running opposite. Up here, it's my 3' end at the top. And here, it's my 5' carbon. Now that's important because when new DNA strands are built, the DNA can only be built in a 5' to a 3' direction, which means if I was building this right side of the strand, I would have to build in an upwards direction from the 5 down here to the 3 up there. Whereas if I were building this side, I would be going the opposite way. I'd be going top to the bottom because I would be running 5' here to my 3' there. So DNA is what we call anti-parallel. In other words, the 3' side on one end is matched up with the 5' end on the other side. They're running opposite. And you can even see how this sugar molecule is upside down compared to that one. They're running opposite directions. Now this is important because if you can only build in one direction, this means as we're trying to split it open and copy it, that's not going to work out perfectly for us. So you can see this one is splitting right up the middle here. And since this side is going 5 here to 3 that way, it can just build all the way right up and through. Whereas this side starts and builds, and it has to start over and build, and start over and build, and start over and build, and it's about to start over again, because for this side, it's going to go from 5 here to 3 there. So we call this one that can go right in the correct direction our leading strand. This one that's having to start over and start over and start over, we call that our lagging strand, because it is not going in the same direction as the rest. Now there are a lot of enzymes that have important roles in doing this. And the first of these enzymes is what we call helicase. Helicase is the enzyme that causes the entire thing to start to unzip and open up. And it would go right back together again if it weren't for these little guys. We call them single-strand binding proteins. And what they do is they keep the entire thing from just bonding right back together again and shutting up. There's no way we're going to be able to add the other side if that doesn't happen. A third important enzyme going on here is this one up at the top. This is called topoisomerase. And topoisomerases are a group of enzymes where what they do is they help they relieve all of the strain that's happening from the twisting. Because if you're just unzipping this strand, and the strand is already all twisted up, as you start to pull it loose, this top part is getting very tight. So this kind of helps it unwind some, so that way it can unzip more freely. So basically now we've got three things involved: helicase unzipping, topoisomerase getting rid of the strain, and the single strand binding proteins getting it separated. Now we're ready to start building the DNA, but there's one problem. New DNA cannot attach if there's not a 3' sugar end available for it to start on. RNA does not have that problem. So there's an enzyme called RNA primase. And RNA primase is going to stick a little piece of RNA on there, and that's going to start us going. So if I look at my diagram that I've drawn over here on this side, you can see I've already unzipped it some. I'm not showing those enzymes that are involved. But if this is a 3' end, this can be my 5' RNA that's getting started. So that would be a C going on here. On this side, I'm going to have to build the opposite way. So this would be my RNA here, and it's going to get going. Now that I've got it started with my RNA primer, I can start adding DNA on it. And the enzyme that's going to do that is one called DNA polymerase 3. So DNA polymerase 3 is going to start building in this 5 to a 3 direction. So it'll put on new DNA nucleotides here and here. Same thing here, 5 to a 3 direction. It's going to start adding new DNA nucleotides here and here, but this one wants to go this way. So as I keep unzipping these, as my helicase moves on, my DNA polymerase on my leading side can keep on attaching brand new sections of DNA. It can keep on going this way. But my lagging strand here is having a problem. It needs to start all over with a brand new piece of RNA primer, and it has to go back in this direction because remember, it's building 5 to 3, but that happens to be over here. And now it hits the end and it starts over. So it can keep unzipping all the way up. My leading strand can just keep going and going and going, just like that. Whereas my lagging strand, once again, has to use the RNA primase, put on a brand new piece of RNA, and then go again until it runs out of space. Because it's going 5 to 3 here. So that's making this my new 5' end here. This is my new 3' end, and these are my 2 new strands forming. Now, once the whole thing has been unzipped, in the spot where it's doing the unzipping, we call that the replication fork. It's the main spot it's unzipping. But it is not done until we get rid of all of this RNA and join up all these little chunks that we call Okazaki fragments. That's the chunks that are made on my lagging strand. So another enzyme is going to come in. This is DNA polymerase 1. And DNA polymerase 1 is going to get rid of all of my bits of RNA, and it's going to replace it with pieces of DNA. So that A becomes a T, this G becomes a C. There we go. So that is DNA polymerase 1. At this point, the strands are almost done, but the only catch is these are not joined. So our last enzyme is DNA ligase. And DNA ligase is going to seal up these last bits. So those are the final enzymes here. Helicase unzips, single-strand binding proteins keep it apart. Topoisomerase gets rid of the strain of twisting. RNA primase attaches those RNA primers to get the replication going. DNA polymerase 3 copies the DNA in a 5' to a 3' direction. DNA polymerase 1 replaces the RNA with DNA. And then last of all, DNA ligase joins together all of the Okazaki fragments that are on the lagging strand.